Okay. Okay, so let's start this thing. Oh, he's here. So if you want to make any intros, then feel free. Yeah. So you want to say anything or not? Okay, he's done. Okay, so the plan for the 3 days is we'll go today through clickety thingies with Cytoscape just to get you going with getting some sort of data in and then annotating it and then making it or trying to make it look pretty. Then tomorrow it's gonna be about where the data is coming from, how it's generated, how to deal with it. It's actually the most messy part. The protein interaction data is pretty diverse, pretty complex, and then you've got to make choices what to take, what not to take, what things are ignored because of the way Cytoscape works. And then on the third day we'll try to use a couple of extensions, so-called apps in Cytoscape to get data in or to do some sort of data analysis or looking at the data. Okay, so if you've got, so this is roughly the outline for the next 3 days. Oh, by the way of introduction, I'm a staff researcher who's been here for years. I'm located on the second floor here in David Eisenberg's lab, but I'm also associated with Matteo's Pellegrini's lab, so it's pretty complicated. And my technically main interest is running the data of interacting proteins. It's a database of protein interactions that's been started here probably in 1999 or so, and I've been dealing with this thing since probably 2004 or so. So that's my connection with the Cytoscape, and so I'm on a side that produces the data, or not produces, it takes data from papers, converts it into some sort of database records that then can be read into Cytoscape. So that's where I'm sitting there. Otherwise, by training, it's hard to tell. I got a PhD from UCLA from Wayne Hablow's lab in molecular biology program, but it was mostly biophysics, whatever. So technically, by training, I'm experimental biologist. Make it more entertaining. Okay. So anyway, that's how Cytoscape looks like. You can get it going by going to cytoscape.org. It's relatively well maintained as long as the main part of Cytoscape goes. This thing is updated pretty regularly, so every now and then you're gonna see information that this thing got updated, and then you can install new version and keep it going. Cytoscape is academic program, so every now and then things might not work, or they don't work for years, and then that's situation with academic software, but it's way better than anything else for drawing protein interactions or networks of things in general. The data model that Cytoscape deals with is a graph. So essentially, whatever you've got in your experiments, in your data, is getting reduced to a bunch of points connected by lines when you draw it, and technically it's collection of pairs of vertices connected by length. And that's all that's there in terms of data structure. You can attach a bunch of labels to either nodes or vertices. Those two things are names are used interchangeably, and you can attach a bunch of thingies to edges. And then some people distinguish between graphs and networks by saying that graph plus attributes sitting on nodes and edges is a network, but I mean, it's pretty arbitrary. Depending on application, those lines might be unordered, so there's no distinguishing between where edge starts and ends, or it might be that in some applications those edges are directional. So that's all that's there in a data model. There is a tons of math behind it, and there are a couple of boring books that deal with the math behind it. And because there's a lot of math, there are parameters of the networks that are derived or introduced by mathematicians in more or less abstract way. Some of those things are commonly used, some of those things are less common, but they do exist. If something strange pops up, just use Google and try to find the math page about it. I just gave a pointer to relatively old popular science book about networks, and there's quite a few of those you're gonna find on Amazon or wherever. So those things are much more readable than whatever is here. And the biological context is pretty well described. I'll move this thing away. Under this link, this is an online course run by EBI in England, next in Kingston, where they describe provide a lot of information about biological interpretation and of networks. So that's the data model, that the data coming from experiments got to be squished in. Oh, come on. And depends on application, the way of squishing things into the data model varies. But every time those three things do have to be taken into account, that all the interactions are binary. There's edge connecting only two nodes. There's a more abstract way of graphs, something called hypergraphs, where you can have edges connecting three or more things, but Cytoscape don't do the hypergraphs.
I'm in the middle of class. Trying to set up something really quick. Sorry.
Okay. I'm gonna ask him to get off. Okay, so no matter what you do with the data, you've got to remember it's got to be made into binary, into pairs of proteins interacting. You can't make 3 or 5 interacting at the same time. The network, as shown here, corresponds to one time point. So this is one snapshot of whatever is happening at a given moment of time. So there's no time dependence in those models, and there is no spatial information. Positions of the nodes are mostly determined by how pretty this thing looks. So there's nothing there in terms of position. Otherwise, you can do whatever you wish. You can assume that the node in the graph is corresponding to one gene, or one protein, or one protein state, or one protein domain, or one protein molecule. It's up to you. The most common are the two versions here. people tend to think about the node here as corresponding to one gene, let's say actin or myosin or P53 or whatever. Or they might assume that it's just one protein, so P53 protein or actin protein or whatever. Those two things are not identical because some genes make more than one protein. If proteins are spliced, then you're gonna have one gene producing 2, 3 more variants of proteins, and depending on applications, you might or might not care about it. So there's some choice to be made when you're dealing with mapping your molecules or protein or whatever to the nodes. Same thing goes with interactions that the edges are representing. Those could be interactions as you think about them, two proteins getting stuck to each other. And you might treat this thing as interaction, but there are more abstract way of thinking about it, about interactions. There are so-called genetic interactions where you look at how phenotype is changing when you mutate one protein or the other, and depending how additive the effect is, you might infer that those two proteins are somehow affecting phenotype in a codependent way, and then there's this genetic interaction there. It has nothing to do with physical interaction between two proteins, but it might be represented as an edge. Another way of making connections is by so-called functional linkages. You might connect in your graph two proteins or two genes that function within the same metabolic pathway or regulatory pathway. Then they also do not have to interact directly with each other. One might be at the very beginning of a pathway, another one might be at the very end, but they function in the same pathway in a cell. So then some people might connect them, saying that their function is very similar. So let's connect them. So this is another way of marking of using edges. And then you might have experiments where you measure correlation in expression between different genes, and then if two proteins are expressed in a coordinated way across different conditions, then you might make a connection between those showing correlated expression, and it's gonna look like an edge, but the meaning is once more different. Each of those edges might mean something different, so it's worth to be aware of those things, and you've got to know what you're dealing with essentially. So now the problem is that we've got to make only connections between two nodes at a time. And this is not what experiments are returning in a number of cases. In some cases, yes, if you do something like ACE2 hybrid experiment where you've got two proteins and then you put them in the same cells and the gene is turned on, then yeah, they interact and you've got only two things to connect. But you might co-purify an entire big complex by let's say gel filtration, identify 5 proteins there, and then you're gonna end up with entire group of proteins coming from experiments as interacting within the same physical complex. Cytoscape doesn't allow you to put it into the model which is based on pairs. So this huge entire complex have to be somehow converted into pairwise interactions to be able to enter into Cytoscape. Another class of experiments is where you've got one protein tagged to be purified by affinity on the affinity column, and then together with this protein you might have several co-purified together. This is going to be reported as co-PI or pull down or whatever, and you're going to have a bait protein purified with, let's say, 5 or 6 or whatever praise. Once more, experiment is going to give you all of those, and then this list of proteins got to be converted into pairs, because otherwise Cytoscape or any graph-based method is not gonna take them. For the first case, for purifications where you know that all the proteins are within the same complex, the proper way would be to connect every two proteins by an edge, because then it means that each pair of proteins is within the same physical complex. And you can do so-called matrix representation of physical complexes. But those are only situations where you know that all the proteins are at the same moment of time in the same complex. And depending on experiment, you can or you cannot say that. In particular, you cannot say that for affinity purifications. For affinity purifications, even if you purify only one bait and two preys, there's a chance that you've got two preys competing for the same binding site on a bait. And then, yes, list is gonna be three proteins, but you can't tell that two preys are interacting with each other. It could be that either one or the other is interacting with the bait, but there's no interaction whatsoever between the preys unless you demonstrate in some other way. Because of that, affinity purifications should be converted into binary complexes according to so-called spoke model, where a bait is connected only with one prey at a time. Other preys are connected also one at a time with the bait, but there's no connections made between the preys. Affinity purifications never tell you that two preys are interacting with it. They might be competing for the same binding site on a bait. So there's a lot of confusion in the papers, in literature. People are initially doing matrix expansion for everything. And then suddenly someone who was more informed than bioinformaticians who know nothing about biology, that, oh yeah, it's not really true. So then people started to convert things using spokes. For affinity purifications, for physical complexes, there's still some cases where people expanded just as a spoke in arbitrary way. Some people expanded as a matrix. So if you're getting some sort of data set, it's reasonable to see, or at least try to see how those experiments in there were converted into binary interactions. So that's something to be aware of when looking at different networks. Okay, so as long as things that you can hang on as attributes on nodes and edges, for every graph you can run in Cytoscape some sort of analysis of the network topology of connections, and this thing is gonna put you a lot of numbers to attach to both nodes and edges. They are mostly derived from those mathematical from the mathematical side, from the graph theoretical side, and those things can be used then to somehow infer something about individual proteins or edges, how important they are, how do they fit within the larger network, and things like that. Otherwise, there are pretty obvious things you can put there. You can attach to each node name of the gene, name of the protein. You can attach identifiers from different databases. The number of databases keeping track of proteins and genes, and each of those might have its individual identifiers. You might hang all those things on the nodes. You can attach gene ontology terms or any sort of annotation that tells you something about function of the protein or localization within the cell. So this is the way to sneak information about where individual proteins are observed. You can attach numbers coming from your experiments. This is, let's say, expression level in a given experiment, or methylation of a given gene, or whatnot. So all those things can be hanged on individual nodes, and the same thing goes for edges. That you can attach just those things, numbers coming from the analysis of how things are connected within the network, but you can add information about what interaction type you're dealing with. So is edge corresponding to direct contact between two proteins, or just present in the same complex, or is it a genetic interaction, or is it just correlation expression? So we can hang all this information on individual edges. So that's what can be put there. And all that Cytoscape does is just letting you collect all this data, and then make the graphs corresponding to your data look more or less pretty, or at least informative. So that's how it goes. On the rest of the slides, there's some information what you can do with Cytoscape, just so that you've got some sort of a log of what we're gonna go through. But I will actually stop PowerPoint and go back to Cytoscape and try to play with it. This is probably best way to learn how to use it. So let's stop for 5 minutes, and then we'll go to Cytoscape. Okay. Are there any questions? Any questions? Any questions online? Let's see. No, there's no questions online. Okay. So let's get out of PowerPoint. Okay. And we'll make a 5 minutes break, and I'll just start Cytoscape. Okay, so once you've got Cytoscape installed, and its startup is relatively slow. Cytoscape is huge and modular, and takes a while to start. Come on. But ultimately, you should see something like this. Okay. And this is essentially a collection of no changes. No, cancel. Block some. Okay. Whatever it is. Hopefully it's not gonna hurt me. Okay. At least not today. Okay, so this is essentially a collection of 3 panels. If you really want, I think they can be, well, they can be made bigger, smaller, and some of them can be popped out of the windows, but nonetheless, there are 3 things. There is the main panel here. This is where the network is gonna show up, and initially you're getting the welcome screen with a couple of networks that are preloaded there. I just click on this thing because it's useful to have something there. Then below it, there is a panel that provides information either about nodes or about edges in a network. Okay, this is essentially a table with a list of attributes hanging off the nodes or edges that you can attach, or you found when someone gave you a network. filing in Cytoscape, and the same thing goes for edges. Then you've got one edge per line and then bunch of attributes there. And then the third panel here allows you to modify networks and change the way they look and scale them and so forth. Okay, to navigate the network, you can just drag it somewhere with the left button when I move around. Okay. If network doesn't fit, then the panel in the lower right corner shows you where within network you are. So the marquee here corresponds to the area you see in the screen, and you can drag it around and go around entire network. If you don't need this panel, you can click and it's gonna go away or reappear as needed. Now, if network is too large to fit or too small so you don't see it, you can zoom in or zoom out using either buttons or you can zoom in with the scroll wheel. That's how it goes. Note that this is not changing relative sizes of elements in the network. That's right. This is like getting closer or farther away from the network, but not making two nodes getting closer. So we're not changing anything with the network. It's just moving around and looking through a magnifying glass. But nothing is changed. To modify the network and scale it so that things are spaced farther away or closer, you can use the layout tools on the left bottom, and then if you click here, you're gonna have a panel showing up with two sliders. The slider at the top is gonna make the network bigger and smaller, and you'll see that this is different than what we had before. Now, if we check here, now the edges are becoming, where is it? My thing is, oh, here it is. Things are getting, if I go down here, see now it's very crowded because I zoomed and make things small. So distances between nodes are getting smaller, but the size of the nodes is not changing. So they start to overlap. If you go up, they will go expanded. If you want to go farther and the slider doesn't let you, you can always click here. It's gonna go back to center, and then you can keep making things bigger or smaller. And here we're roughly, oh, maybe not. Or here we're roughly as we had before. Oh, as you've noticed, when you click on an element of the network, either a node or an edge, it's gonna, well, depending how lucky you are. Yeah, with edges, it's tricky. If you want to click on, but you can always press Shift and drag to go over elements and select more than one. Okay, and then if you start dragging those things, you're gonna be moving one element, one node at a time. So you can do this thing. You can, if you want to move groups of elements together, you can, like here. Here, okay. if you want to move entire group of things, yes, you can. You can move entire group after selection, or you can use layout tools here. And then if you have the selected only checkbox on, then you'll be scaling only things that are selected, right? So if we select, let's say, things that are those three things, then we can move only those three things. So another option is to have things that are selected rotated with the lower slider. So then that's what it does. If selected is checked, then you're gonna move things that are selected. If this thing is not checked, then entire thing is gonna rotate. So that's when you're manually play with the network layout. It's all about creative selecting of things that you want to move around, and if you want to move one or two or three nodes, it's relatively easy to select those things by clicking. If you want to make selections in a more creative way, then under select there are options to select things, either edges of or nodes in a some sort of a systematic way. And let's say if we select this guy here and we want to select things that this guy is connected to, I think you can go to select nodes and you can select first neighbors of selected nodes. So we're gonna select everything next to this guy. Okay, and if we want, we've got the choice if we want to ignore the directionality of the edges or not. So we can always go to select nodes, first neighbors, undirected. This is gonna select me everything next or up one. So now I can either drag all the neighbors around, or I can rotate only them and nothing else, or I can scale it. So it's useful just to check what those different options for nodes and edges are. So it makes you work faster essentially, and reduces the amount of clicking that you've got to do. Sometimes those things hiding here might be useful. So that's basic manual modification of individual things or regions of the network. Apart from dealing with one or few nodes at a time, there's a layout panel which allows you to rearrange things according to some algorithms that people came out hoping that those things will generate nice graphs, and there's a plethora of those things, and the only way to learn how they work is to try and see if it even works or not. We can try, let's say, the circular thing, and this is apparently making circles of things according to some sort of algorithm. It's hard to tell what it did here, but that's what we ended up with. It's gonna work better with a smaller network. This one was way too large to actually test things. Mm-hmm.
So where did you find information on how this to make have that shape?
I don't know. Okay. That's the problem with some of those prescriptions how to arrange things, that some of them are are more obvious. Some of them are not, and that's why I'm saying the only way to figure out is by trial and error. It's literally trying if any of those things work. Some of them are relatively obvious, but I go back to this thing on a smaller network because this is a hairball that's hard to work with. I think the original layout was probably done by hand. That's my guess. That some the very common theme is you apply one of those layouts initially to get something roughly reasonable, and then you start to tweak it by modifying regions by hand, and this is more of an art than science. It's like with Photoshop or like with Illustrator. I mean, the only thing I can teach you is what to do to perform one operation at a time. The rest is experience and how much artsy you are. Essentially, some people are better than others, and some people's got more experience than others. So there's this is the artsy part of Cytoscape. Some people are really good, and some of those networks look really nice. But it's hard work. It's not like it's gonna come up out of nowhere. So now let's abandon this thing and let's try to get something reasonable, something smaller for us. So to get rid of this thing, we can always close it. Cytoscape is gonna ask you if you want to save the session or not. At the moment, we're gonna ignore saving, and we're gonna continue, and we're gonna end up without anything at all. And then let's try to get something in. Okay, I prepared two files here. Let's get this one first. Those are regular text files. Okay, I'm gonna open it with this thing. No, file, go away. No. Okay, I'm gonna get rid of no. Get rid of no. File, open. And let's go. This is desktop. Those two files or three files are and PowerPoint are on a Google site. I send you links, so you should be able to get to those. There should be. I just got them here, and that's one file. Okay, it's coming from the I've got to listen to everyone talk on Thursday. One of the structures coming from cryo-EM for the polycomp complex bound to nucleosome, and I just converted the structure into a file format that Cytoscape can read and interpret as a network. Essentially, the way the file looks like is the strictly required are first three columns. The first and second and third position, so here and here, those two things, those are identifiers of the nodes. They can be anything. I tend to use for proteins identifiers from the UniProt. So then I can correlate the node with information that's pulled in from UniProt. So the first 9 is interaction between protein Q15022 and protein Q92833. Okay. The second column is so-called interaction type, and in the canonical representation, Cytoscape uses PP because it's interaction between protein on the left and protein on the right. For other molecule types, it might be different, might be DP if it's DNA protein, or RP if it's RNA protein, or things like that. So those are conventional ways initially Cytoscape was using. Different data sources might be using different interaction types and different meaning here. So this creates sort of problems when trying to merge data or use different apps. But I tend to use the canonical thing without putting much meaning behind the type besides the type of the molecules interacting. The fourth line is redundant. I do use it to convey how many atoms of this protein are of how many atoms are between those two proteins are close or not atoms. It's atoms. Yeah, I think atoms residues. This is, well, it's a number that corresponds roughly how close those two proteins are in space. So if the number is large, it means there's a large number of atoms from one protein next to the other one. So that's about it. But this is completely redundant. Not necessarily for specifying the connectivity of the network. To get this file in, we go to File. Okay, we go to Import. We're importing network from File. Okay, we've got to go to Desktop W2, and we go to our file. We open, and now we're gonna have a panel that allows us to choose what's the meaning of the columns. Okay, I'm gonna ignore the last one. To ignore it, I'm gonna, well, first thing I've noticed that the first line in a file got interpreted as a name of the columns. This is not what I want. The first line is also interaction. So I've got to go to advanced options, and I've got to uncheck use file first line as code. I don't want this. So we start from first line. So let's do okay. So now we've got just default names of the columns, and I'm gonna ignore the last one. So I'm gonna click here, and I'm gonna say not imported because I don't want it. This is something that came extra. We don't want at the moment. We're gonna specify that column 1 is a source or beginning of the edge. We're gonna specify that column number 3 is the target or end of the edge, and the column 2 is gonna be interaction type. Okay, as you see from this thing, the edges in Cytoscape are directional. Each edge does have a beginning and an end. If you want to ignore directionality, the operations performed on the network will have an option to either ignore it or not. So this is not a problem in a sense that edges can be treated as undirectional, but internally they're always directional in case it's needed. So this is something to be aware of. And then once we click on OK, here's our toy network. Okay, and we can make it sort of crowded, so we can go to layout tools and scale things up, and now things don't overlap. Okay, and if we want to rotate the whole thing, we can rotate this thing, and here's our hairball. So as you notice, we can have nodes connecting this starting and ending at the same, I mean edges starting and ending with the same same node, and we can have multiple edges connecting the same pair of nodes. Mhm. So this is what came out from the file. Okay. Let's see. Now, this is pretty uninformative because no one knows except for few Uniprot and aficionados what those stickers are. So it would be useful to attach some sort of informative data to at least our notes. To do this thing, we've got to go to Uniprot. Okay, so let's go there. Let's go to Uniprot. Okay, this is the website. They changed. They changed the interface within the last half a year or so, and I'm still lost on the old one. But information is the same as far as I can tell as it used to be. And for individual proteins, one can always select identifier, control copy, accept, and search, control V, and search what this thing happens to be. Uniprot should be able to find it. Okay, provided it's alive. I have, oh, I've got by accident this thing. Yeah, so so here's information what the protein is, and one can painstakingly take one identifier at a time, check what this thing is, and data to Cytoscape by hand. It's tedious when it's more than one or two proteins. So there's a better way. We can always go to, I think, advanced. No, we can go, they move things around. We can go to ID mapping. Okay. Then we've got to get the list of all the identifiers here. This is tedious to get it from the file here, but we can use Cytoscape, and we can go to node table. Okay. And then we do have so far just one column with name. If we can select, we can see, I should be able to copy all the identifiers from the column. Okay, and no. Well, if not, I can select everything and Control C. Control V. Yes, I can. I can, just like in a spreadsheet in Excel, I can select everything here. Control C and Control V gets me identifier here into the search window. Okay, and then I know those are Uniprot identifiers, so I just select this thing, and this is gonna get me just identifiers. So I don't. And let's try to map. And let's see what we've got. This is gonna get me Yes, this got me identifiers here and corresponding entries from the UniProt. Okay. If we want to save the results as a file, we can download things, but before that we can customize columns to see what we need. And there's a lot of things that can be imported. Let's see what's useful. Okay. So let's get entry name. Let's get not names. I just want primary gene name. Okay, so that I've got one, not many. There might be synonyms. Well, I can get synonyms. I'm gonna get organism ID. Why not protein names? Okay. Sequences. No, I don't. Oh, length of the sequence is probably okay. We can use it later on. But there's a lot of extra stuff that can be imported. Miscellaneous, what's hiding here? I don't care about this thing. Gene path, subcellular structure. Okay, so let's close it. So this is what we're gonna be getting. Okay, and then once we decided we've got the information we need, we can always go under download. We want to download as a table. Let's see. Text. Well, let's try as text. Compressed, no. And we download everything. Okay, we can preview if it looks okay. No, text is bad. We're gonna get RDF, not lift TSV. That's better. If not sure, we can always preview. Yeah, this is tab separated. So this is what we need. So if it's okay, then we're gonna download, and I'm gonna put it into W2. Okay, and let's name it something reasonable. Unique rot no txt, or let's call it tab. Okay, save. So let's see, what do we have? Here we go. Now here's our file. I can open it in the editor. That's the one that I pulled in. So this is our original identifier. Okay. And then a bunch of fields that was added and pulled in from UniProt. So this way you don't have to do those things by hand. And there's column for entry name, protein names, organism length, gene name, primary gene names. So list of synonyms and organism ID, and probably that's it. Okay. So now how to get it into Cytoscape. Okay, so now we go back to Cytoscape, and now we go to File, we import, but now we're not importing network, we're importing table from File. Okay, we do all files, so that's our file. We try to open it. And now we've got another panel that lets us modify how the file is going to be interpreted. Okay, this time we want to keep the checkbox keep the first line as a column names, because that's how file look like. So here we've got different column names and corresponding information down here. One of the columns in the file do have to match identifiers of the nodes here, or at least previous attribute for the nodes. The easiest thing to do is to make sure that one of the columns in the file we're importing match the identifiers of the proteins, and we can use either from or entry. Those two columns are identical when they came from UniProt, so we can keep it entry as our key. And then when importing, Cytoscape will match the identifier here with identifier of the corresponding node. Let's say this one is gonna be matched to this one. So all the information in a file in a line with P62799 is gonna be attached to here, provided we say that we want to get information in or not. So let's keep entry name. This is name of the record in a UniProt. Protein name is long, so in context of Cytoscape, it's not gonna be too useful, so we can always ignore it. Okay. What else do we have? We've got organism. This is human readable name, so let's keep it. This is protein length here. And this is a number, so we can make sure that the value is going to be interpreted as a number, as integer. And Cytoscape guessed correctly that there should be numbers. It also guessed correctly, let's say here, that this is a string, that this is piece of text in an organism column. So attributes can be either strings or integers, or floating point numbers, or lists of those. So depending on what's in a given column, what's needed, one of those things can be selected for a column. The values in a column for all the nodes got to be the same thing for the edges. The attributes got to have the same type for all the prods. So you can't mix numbers with strings, let's say. So we've got organ length is okay. Here's our primary gene name. Okay. Synonyms, we're gonna ignore them, or for the experiment's sake, we can actually say that those are actually list of strings. That's right, because we've got sometimes one, sometimes two, sometimes three. If we want to get all of them, we can say it's a list of strings. And then we've got to specify how they are split into bits and pieces. And in our case, it's gonna be space. That's right, because there's nothing here, and columns are separated by tabs. So this hopefully should work. Okay. And then this is numerical identifier of species coming from CBI. So let's get it in. Okay, so we've got everything in. So then do okay. And let's see, did it get in? Well, yes, we do have now node table with more things hanging in there. For the gene name, we do have values, individual strings, individual gene names. For gene names, for synonyms, we specified we want to get the list of things. So now we do have either nothing or a list of things. Lists are represented by a pair of square brackets with commas separating the values. So some things came in, some things are missing. Let's see what's missing. Why we don't have this thing, I think I know. And I also know, but we'll get to this thing in a moment. Okay, so did you get this far? Okay, I see two nodding heads. Hopefully, third one is also nodding. So that's how you can get in a relatively straightforward way information about proteins from UniProt, providing you do have your nodes identified by UniProt identifiers. If not, and it might happen, you might have something else coming in from the data or from the from collaborator or whatever. Then you can try to see if you can convert identifiers used in your data set can be mapped to UniProt. And then you can hang the UniProt identifier as the value of one of the attributes for your nodes, and then you can use this thing as a key when getting information from UniProt. And the mapping service is pretty extensive. You can map different things. There are identifiers coming from a gene bank, from EM, from NTRES, from RefSeq. So this thing takes time and to find out how to do those things. This is the major bane of bioinformatics, mapping identifiers. This is a separate issue that's not related to Cytoscape. It's just mapping one data set to information from some database and trying to find out how to match identifiers. This is a large part of the bioinformatics, just fixing messed up identifiers systems. So that's sad reality, but UniProt allows you at least to try to do it in a clickety way. In some cases, you will have to resort to scripting, and there's no way to avoid it. In some cases, it's easy. In some cases, it can be really messy. So that's sad reality. But this is the initial try. If this ID mapping in UniProt works, then saves you a lot of time. More about those things tomorrow, because those things are coming together with other issues with the data sets. So that's how to get your nodes annotated in a simplest possible way. And this is probably one more thing. Once you get your initial data in, and every time there is a major step in data processing, like getting new annotation in, then it's wise to first start saving things. Okay, so you can first just save as, and you're saving so-called session. Okay, so let's call it, how to call it? Network 1, okay, and save it. And once this thing was written, we can close it, okay, and then we can go open our network 1, and we're gonna retrieve everything that was in Cytoscape at the moment we save the screen. This includes not only the information, what are the elements of the network, what are the nodes and connections. It also corresponds to all the layout, everything is saved. The state of the Cytoscape is saved as it is. And it's reasonable to save this file every now and then, because Cytoscape every now and then crashes. It happens. It might happen during this workshop that this thing is gonna start misbehaving, and then if it happens, and if you can't help it, but you've got to kill Cytoscape, restart it. If your session hasn't been saved, then you're gonna lose information between the time of the crash and the last save. So it's reasonable to save it every now and then. just for the safety. There's Cytoscape doesn't do backups or anything like that on some way, so it's in your hands. Okay, and then there's also a chance that you will perform operations that can be unwound. Cytoscape does have a little bit of undo capabilities, but those things not always work, and they don't always work the way you think. So there's a chance that you're gonna perform some operation that you can't unwind, can't fix if it's not doing whatever you hope it's gonna do. To avoid problems, avoiding wasting time because of those things that you do that can't be that destroy useful information, it makes sense to every now and then make a clone of the network that we've got here. So this is done by going to new network and doing clone current network. And now we've got initially two identical networks. They might be drawn slightly differently, apparently. That's what's happening, but those two networks are the same. And then one of them stays as it is, and then we can play with the other one, and they are, as you see, independent. So one of them serves as a backup in case something goes back, also helps you to keep track of what you do, you know, at what stages of data processing analysis you are for each of those clones. And then you start modifying the new copy. And this way you can keep track of what was happening. One of the issues, at least I've got with Cytoscape, is that it's very hard to reproduce whatever was done, because this is mostly clicking. And this is very hard to document. So at least if you make those clones of the files and scribble down somewhere what they correspond to, it might be easier to document the flow of your work. So that's another part on the safety and how to avoid the frustration because something after 3 hours of work went up in a crashed program. So those are two things. Okay, another break. So let's oh, there's something in chat. Let's see. Where's chat? Where's the chat? Can't see the chat. Chat disappeared. Where's chat? Oh. Where's chat? I did hide it somewhere and I can find it. Where's my chat? Oh, here it is. Oh, there is a question here. Let's see. Could not hear the question. I had trouble in Turbo as GZ. Oh, as long as importing tables from the UniProt, there's an option here when importing to import as compressed file. If it's importing as .gz, you've got to uncompress it on the receiving on your laptop. So that's or import it as as just table, not the compressed one. So that's how it works. Let's see. I'll try to reproduce the problem. Let's see. So if we do download, compress the yes, let's do this thing. If compression is a problem, you just select no, and then file is gonna come in as a plain text file. If it's compressed, then if we download, and let's get it with the full name. Then it's gonna come in as a compressed file, and then if we open, then there might be problem with uncompressing it. For small files, you can import this thing as uncompressed. You should be fine. If it's large, then you've got to find out how to uncompress it on your particular computer. Let's see. I don't know off the top of my head how to get stuff out with the Windows. I'll get back to this thing tomorrow. I'll see how to do those things on Windows. I know how to do this thing on Linux. I'm primarily working on Linux, so I don't run into those problems here. So that's how it goes with the compression. For small files, it doesn't matter. For large files with the thousands of proteins, then does matter, and then importing compressed files would be better. So, but I'll get back to you tomorrow. Let's see, and let's pause recording. Okay, so let's keep going. Okay, so here's our network, this time with a bunch of things hanging off the protein nodes. As you've noticed, maybe if when I click on and select one of the nodes, I see only information of this particular node showing up in the table. If I've got more than one, then I'm seeing only things that are selected. This is because the default option for the table is so-called auto mode, which means that if there's nothing selected, we've got all the nodes listed in a table. If something selected, only selected nodes are shown in a table. If you want to see everything all the time, then you can switch to all. Okay. And if you want to see only selected things and nothing else, no matter what, then you can check selected, and then with nothing selected, you're gonna see nothing here. So the convenient thing is auto, but you might have different requirements or so you can modify this thing. Okay, so now first, those things are very uninformative. So let's try to, instead of showing UniProt identifier, the name of the node, let's try to replace it with the name of the gene. Okay. So this, we've got to start playing with styles. This is the collection of settings that determines how you see the network. Okay, and you get to it by going to the styles tab, and there are going to be two options. There should be option for nodes, and we see options for nodes, and then there's another tab for edges. Those are how edges are shown. And then we've got finally overall setting for the network. This includes things like a background. We can, instead of default white, we can select something different. Let's make it, I know, let's make it somewhat different. So set a network, and then background is not white anymore. Okay, but going to nodes, now we're gonna see a lot of properties of the nodes that we see on the screen. Okay, there are some of them are shown by default, and then there's additional things hiding under properties. And if you want to use less commonly modified things, you can select something here. But let's stay with defaults for a moment. And now some of the things are the same over and over and over again for all the nodes. So those are default values that are used. Okay, and let's say that's why we see all the nodes being blue, because the fill color for the node is set to be blue. If we want to change it, let's say to orange of some sort, we can do okay, and we change the color of the color of all the nodes at once. Those default values work if all the nodes are supposed to be rendered using the same setting. For the stickers, we don't. For the stickers, we want to have different stickers for different nodes, and each sticker is supposed to correspond to a value coming from the gene names column. To do this thing, we've got to use so-called mapping. Okay, and it is used for the label now. This is label. So if we click here, we do have a column, and this is at the moment name column, and that's why we see the identifiers here. But this is not what we want. We want to have different columns, so we just click on name, go to gene names, primary, and some of the nodes do have stickers and some of them don't. We'll get there in a moment. But for those that do have stickers, we do have something that at least to geneticists is more informative than Q15022. So here are our gene names, okay, and the mapping that's used is so-called pass-through mapping. There's more than one type of mapping things. In the simplest possible case, we take a value from one of the columns and we pass it without any modifications as the value to be used as a label, right? We don't modify the value in a gene name, you just use it here as a sticker. So this is the simplest possible case. There's more mappings. We'll get to those just in a moment. Okay, if we don't like, okay, so now why do we have things here missing? Okay, well, it's because we didn't get those things imported. Okay, and there are different reasons for that. Okay, one is here. If we look at things that got imported, the P6298 is a total failure. That's right. We've got nothing here. Let's see why it happened. Well, we go to UniProt and we'll check what's the identifier. Okay, this is what this is. P62988. If we go here, P62, come on. Does exist? No, not here. Let's search here. P62988. Come on. Okay. P62988. Search. Okay. Yeah. Okay. So So what do we see here? Okay, it says here the entry is obsolete since a decade, okay, but it used to be ubiquitin, human ubiquitin, okay. This is typical problem when dealing with interactions of ubiquitin. It happens that there are two molecules of ubiquitin in the structure. Ubiquitin is a protein that's not made as a not coded by a gene corresponding to this short 76 or so residue long polypeptide chain that is swimming in the cell as ubiquitin. Ubiquitin is made either as a concatenation of 5 ubiquitins translated all at once and then chopped into pieces, or as a fusion with ribosomal proteins. This is common between for eukaryotes. It does make biologically sense. The fusions between ribosomal proteins and ubiquitin make sense because you make ribosomal proteins when you want to increase protein synthesis, and then it's likely that you will want to degrade those proteins later on, and ubiquitin is used for regulation of protein synthesis, protein degradation. So that's why some ribosomal proteins are expressed together with ubiquitin. This is preemptive way of making enough ubiquitin to degrade whatever you made. Plus, there's a lot of ubiquitin to be made, so you're making 5 copies of ubiquitin per one run along mRNA. So that's why you've got fusion. So this creates problem for annotating interactions with ubiquitin because someone not too clever in UniProt removed the entry for the canonical 76 or so residue long ubiquitin. And this happened 10 years ago. So interaction databases tend to use those obsolete ubiquitin identifier for human, for yeast, for whatever species, so that they refer to one ubiquitin chain, not this concatenation of 5 or not the fusion with ribosomes. And then it creates problems with getting information in from UniProt, because UniProt then tells you this is obsolete thing, go away. So that's why we do see it empty here, but we can do it by hand. We can always use the panel here to type things, so we can make this thing as P62988. We are gonna, let's call it UBQ human. It's gonna be doesn't let me. Well, length I think it's 76. If I'm not, we can call it UBQ. And then did we recover the name? Yes. So now you see we do have the sticker. The whole exercise was to get the sticker here for ubiquitin. So that's why we were missing the sticker here. And then for the organisms, I happen to know that human is 9606. So those things can be fixed at this stage, or those things can be fixed when you import the file, load the file into Excel and check what's there and fix problems. So this was the problem with one identifier, and now there are 4 more things. Let's see what those things are. If I want to select the 4 at once, I can just select those things. And now, if we want to switch from one selection to opposite selection, the simple way is to go to select and there should be selection, there should be invert, selection, social mouse, should be invert. Nodes, we can do nodes, invert node selection, then we switch. So as you can see, all the selected things here are human proteins. Okay, and this is what the paper is about the polycomp complex from human. But if we go to select nodes invert, node selection, now we're getting frog proteins. This is very common for interactions involving histones, and those are histones, those are core histones from one nucleosome that the complex is binding to. For this type of experiments, it's very common to make in vitro or in E. coli is to make xenopus histones, and this is relatively common in experiments that interactions might be reported between proteins coming from different species. So then one way is to ignore those if we don't care and we hope that there was another experiment made with the corresponding pairs of human proteins, or we can keep them, but then we might run into funny problems. Apparently, UniProt wasn't able to get identifiers for those things, the gene names. So then to fix this thing, luckily the entry name tells you what histones we're dealing with, so we can just fix it. This is gonna be our H4. This is gonna be our H2A1. This is gonna be our H2D1.1. I think that's the variant. And this is gonna be H3.2. And now we've got our stickers for the histone, right? So we made this thing a little bit better. Now, this might be a good time to go back to network, and then we can just clone this thing, and then we'll keep whatever we've got here intact without messing with if we mess up something later on. So once more we go to File, and then we do New Network, Clone Current, and now we've got 3 networks. This is our initial one. Okay, this is the one that we fixed in terms of getting the stickers and gene names fine. And now we've got another one that we can work on. So far this thing wasn't saved, so I'm gonna do File and Save, and then I keep all those things saved on a drive in case something bad happens with Cytoscape. So that's one more step. Now we do have strange things here. Okay, so this is our complex, the polycomp complex. This is a complex that methylates histone tails and participates in gene regulation. And those are all those human proteins out there. And if you check the structure, there's only one copy of a given protein in a complex. So then one copy of this guy can interact with one copy of this guy. So edges are just unique to a pair. You don't have multiple edges connecting things. This is different on the other side. We've got histones here, and we've got tons of connections between in between things. Sometimes we've got 4, sometimes we've got 2, sometimes we've got things looping on itself. Anyone has a guess why? How many copies of histones are there per nucleosome? I mean, the genes are the histones are duplicated. You've got eight protein chains per nucleosome. You've got only 4 chains here, so those things are duplicated. And then the loop from starting and ending on H2A histone apparently means that there are 2 copies of histone H2A, and they are in direct contact. So that's what this loop is. Same thing for histone H3A, okay? And then there are more contacts between other histones. So that's why you see more than one edge here. The way to simplify the network is to either do edit. There's a way to where is it? Show there's somewhere. There's a way somewhere. I don't use things because this is not what I like. Oh, we can bundle edges, I think. No, not this one. Clear. Oh, well, we can try. Let's see. Layout. All nodes and edges. Let's see. No. There's a way. I'll find it for tomorrow. How to hide the multiple edges connecting the same pair of nodes. I'll find this thing for tomorrow, together with uncompression things. But another option is just to get rid of duplicated things. Okay, so to do this thing, we can go, I think, to edit, and we can remove self loops. So this is self-explanatory. Hopefully, it means that we want to get rid of the loops here, but thing to pay attention to is we want to do it only on the last network. We don't do anything to the other 2 things. Those 2 things are archival. So I'm gonna select underline 2, and we're gonna get rid of it. So now our loops are gone. And then we can also go to edit, and we can remove duplicated edges. Okay, once more, only from tab underline 2, only from the last network. I'm gonna ignore edge direction in case this node goes from H2A1 to H32 and the other one goes in opposite. So I want to collapse those 2 things, remove all but one, okay? But I'm gonna create an edge table with the number of duplicated edges. So this number is gonna tell you something about the number of contacts between those 2 proteins here. So let's keep it, okay? And we'll do okay. And here's our network after making things a little bit cleaner. Okay, so now we've got our polycomb complex, we've got ubiquitin, and we've got 4 we've got four histones. So this is some sort of a network representation of whatever structure you had with the full atom model of the proteins forming a complex in determined cryo-EM. This is essentially a diagram that shows you which proteins in this complex are in contact, and you can annotate it in different ways. Let's try to keep styling it. Okay. So far, I was changing default style, but if you want to create your own style and save it for later use, you can always go here. You can create a new style. Let's call it my style 1. Okay, this used the default and killed whatever I had before, so I'll have to redo things. Another option was to copy style from whatever we had, and then we would have something better. Oh, we can do it. We can actually go back. Default is still here, so let's copy style. Okay, name, let's call this one Nile style v2. Okay, so now if I want to go to something else, let's say this one, I can select something pre-made. If I want to have my own, where did it go? Here's my, and here's mine. Okay, so so now, well, what we can do is we can now color we can try to color things according to what those things are. We can color the histones in one way, we can color this complex in some other way, and we can color ubiquitin in yet another way. There's more than one way of doing it, but it's all about using mapping in a useful way. And we can either reuse information about the nodes that we've already have here, or we can add another column. Okay, this panel works as a Excel, essentially simplified version of the Excel. It's rather clunky, but for some purposes it's fine. We can create a new column. Let's new single column with let's say string, column name, let's see, let's call it complex. Okay, and then let's first select all the histones. Okay, and then I'm gonna say here histone. Or nucleosome, actually. Nucleosome. Okay. And then it should be. I can do the same thing here. Nucleosome. Okay. Here, no. Here. Okay, so this is my nucleosome. So now all those 4 things do have the nucleosome set in the complex column. Then for the other guys, we can select those. And then they will show up. They will show up here for the complex. I think it's called PM. I think that's PM here. Now, I think there's an option to copy things to fill the column with, but let's see. Let's experiment. Oh, let's see. PCM2. Third. Okay. Complex. Third. Now let's see how it works. PCM. Yeah. Okay. Now you get the formulas next time. For the moment being, let's call it PCM2. 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 PCM2 only. So now we've got all the things here named PCM2. Okay, so, and then there's our ubiquitin without any value here. Having columns like this with same values for some classes of nodes, let's say for the PCM complex, the polycom complex with one sticker and histone with another one. If you've got 10 different subsections of network, is very convenient because it offers you another way of selecting things. In our case, it's simple to highlight things, select things by using a marquee to get all the complex because things are arranged in a way that is easy. Not always the case. But what you can always do is, once you've got the column with the same names, use same values used for members of the same subset, can always sort on this column. That's right. And then it's gonna bring, if we sort in some other way, things are separated into, they are intertwined, but if you sort on complex, everything is gonna be together. So then we can select everything easily by selecting a block of things in a table and then right click. Where is it? This. It's away. This is a stupid thing. Why is not working? Select. Oh, this should be working. Why is it not? Oh, come on. Oh, let's try once more. There's a way to, well, maybe that's not it. You've got to select here. You've got to select entire row on the left, on the left side. Okay. So now, okay. Something's not working. No. Something's not. Oh, no. No. Select. No. There should be select node from table, and this option is not willing to show up. Why is it not? It worked on my desktop downstairs. That's annoying. I mean, you should be able to get a pull down with select the nodes from table. Or some, yeah. So it should, you should see something like this, and then the selection from the table should be converted into selection in the network. Yeah. So this I've got to figure out what's wrong here. There's something. You're just doing right click or equivalent. Yeah. So I should be seeing it, and I don't. So this way, this is a very convenient way of selecting subsets of nodes without clicking forever or trying to do selections on a panel here. It's enough to have a column that you can sort everything in such a way that things are grouped the way you need them to be grouped. Okay. So this is one reason to have this column with the classes of nodes there, and the other reason is that we can use it for another mapping. Now we can set a mapping of the fill color. Okay, we're gonna use as a column our new thing called complex. And then for mapping type, we're gonna use discrete. We can't use pass-through mapping for color because nucleosome or PMC is not a color, but we can use discrete mapping. And then I made a typo here, so there are 2 nucleosomes here and 1 PCM. And then for each of those, we can pick up a color here. And let's make nucleosome. We can make nucleosome, let's say, I don't know, dark green. Okay. And let's make the typed thing also dark green. I think this is the same. Okay. Hopefully, yeah. So this is our nucleosome shown in green, and we can use PMC. We can make it, let's see, let's make it purple. Okay, and then we can use ubiquitin as a default, but there's another option. If you've got 1 or 2 or very few outliers that you want to color by hand, you can use the third column called bypass here, and then if something is selected, then you'd be able to click on bypass here, and this is the color to be used for this particular node or entire selection that's bypassing both default and mapping things. So we can make ubiquitin, let's say greenish. Okay, and now it didn't change because it's selected. If we unselect, here's our bypass value. Okay, so this way you can encode the membership in different parts of the network, different subcomplexes by coloring things using discrete mapping and assigning colors to different classes of nodes. Okay, what else we can do with this thing? There's so far we're mapping between a string and the property that's exactly the same string, so we did nothing. We converted string to a color. We used the discrete mapping. The one more mapping that can be done is we can have a numerical value that's used to either change color or size or something, some property of the nodes. Let's try to use one of the values in the node table. I think we do have a length of the protein here, and we can use this thing to modify the size of the sticker. To do this thing, we'll change the sticker shape from whatever we've got here to a circle. So let's try it. Well, I've got to hide this thing because I don't need it. The options for mapping are hiding under the twisty here. So if we don't want things, we can hide it. We're gonna change default shape to ellipse. Okay. So now we've got things that are elliptical, and we're gonna log node width and height so things are circles. Okay. Now we've got a problem. Our sticker shows up smack in the center of dots that might be bigger and smaller. So one option is to make those dots start from large enough size to have the sticker in. Another option would be to move the sticker to the side. Both things are viewable. Let's try to do the simpler thing. Let's try to make those things bigger. Okay. So to control the size, we need the size of the node here. Instead of default, we're gonna use mapping. Okay, we're gonna use as our initial value the length of the protein. I could have imported molecular weight and then use molecular weight, but it's another option. Okay, roughly the relative sizes should be the same, more or less. Okay, and then mapping type is gonna be continuous. And now we're going to get the ramp here. If we double click on this thing, it's gonna end up with a pop-up panel. And then along horizontal axis are the values of the property in a table. So at this moment, we're starting from 76, this is ubiquitin, up to 12 or so thousand, 1200 for the largest of the proteins. Okay, so this is the range of the length values. Those do not have to be the same, and typically are not the same as the dimension in a pixels or whatever units that the Cytoscape is using to show those. So 76 apparently is very, very tiny. So 76 is converted to 10, and 10 is too small. So let's see, we can move this thing up until we are large enough. This is very clunky, but okay. Ubiquitin is smallest. I'm just looking for something that is gonna fit ubiquitin in. Okay, and then we can make things that are 1200 or so bigger. Okay, and this is clunky. This panel requires practice. It's very clunky and temperamental, and you've got to play with this thing to get the idea how this thing works. Essentially, you can drag different things. You can move things sideways here. You can move things here, and it's trying to auto scale itself. So it's clunky, but as you can see, we can make things more or less okay. So here's our the rendering of the interaction network that somehow conveys how large those individual proteins are. You see the ubiquitin is tiny, histones are tiny, the members of the polycomb complex are way larger. So here's another way of drawing the network, and we can take this thing once more. I can collapse this thing, and then I can go to network, and I can file, and that file I can do file new network clone. So now I've got this version of rendering of the network saved, and then I can keep modifying it. If I want to change it even more, then I can keep changing it. Every now and then I do save thing, so that I don't lose it if something bad happens. So any questions about how to modify those things? Doesn't make sense? So, and and the same operations can be repeated on the side of the edges, right? So far I was playing only with the different ways the stickers are, the nodes are shown. Oh, if the contrast here is bad, if things are not visible, obviously you can change the color of the label. Maybe if we make it white, it's gonna be better. Yeah, this probably is better. So and besides whatever properties you see here, there's way more things down here, so you can decide which one do you like to see. There might be one that's not shown by default, which is label position, which is useful. If we check here, then we should have label position here. And if we click here, the default is that the labels is in the center of the sticker. This is why all the stickers are shown in the center of the node. If we want to move it somewhere else, we can always drag this thing to a side, let's say like this, and see what happens. Okay, and now the stickers are to the side, but now they are poorly visible, so back to label color. Let's make it very black somewhere. RGB, let's go to very black. Okay, no, this is not what I wanted. I want to let's see some RGB rainbow swatches. This is better here. Okay. So here we've got black stickers, and if the font size is too tiny, we can make it bigger. So here's another version with a different way of labeling things. So and then arrangement can be done either by hand, as I was playing here, or we can try some of the layouts. This is easier to play now than with this big network we had before. If we go on layout, let's see what it does. Let's see. Let's try the one I used before, the circular layout. And this is too crowded, so we can go and make things separated more, and then clicking on here is gonna get you entire thing. So clearly, if network is small enough, you're gonna get all the nodes arranged in a circle. If you apply circular layout, Okay, and there are actually two of those. Let's see, we can try to let's try if we can. There are two flavors of circular, and it might be that at least one of them circular. Yeah, so if there's a selection made, some of those layouts do allow you to apply it to selected nodes. So let's see what happens if we do it here. Okay, so we do have a small circle made out of just selection. So our histones were made into circle, which with four things looks like a rectangle. If it's too small, then we go to layout tools and scale it a little bit more. So you can now, and if we want to rotate it, and only the selection, we can rotate this thing on its own. That's right. Now we can do the same thing with the regulatory complex and apply layout and then circular selected nodes only. It's too small, so we go to layout tools. This is too far to the right, we can't zoom more, so then this is gonna reset the scale to 1 and we can make it bigger. Okay, so here's our complex. And if we want to rotate the entire thing, we're gonna rotate without checkbox, and then we're gonna go with this way, like here. Okay, and we can select stuff here and move it to the side, and we can move this thing here. Okay, so here's another version of the drawing, partially done by hand, partially using one of the layouts applied to the subsections of the network. We can take this thing now and file new network and clone. So we've got now one layout, another layout, and here we've got the clone to play with. We can go and try other layout approaches. The ones up to here, those are built in and provided by Cytoscape. When you go to Y files, those are based on a library provided by a third party, by a company that licensed this thing to Cytoscape. So initially you're gonna see this install whatever layout. If you click on this thing, you'll have to get out of this thing. No, here. You're gonna go to install the layout algorithms, okay? And then if you click on install, you've got to accept license from whatever. There's nothing special there, so you do accept. And now you do have the actual algorithms that you can use, and there's a competing Y file thing, circular, so we can apply this thing, and this is equivalent of whatever we had before. Some of them are useful, some of them are not too useful, but the only way to do it is to try organic. Is yet another one, and then if it's too crowded, you can always expand thing and then rotate if it's not exactly what you want. And so this is some default way. One of the things here generates a layout. The only way to Uh-huh. Yeah, so you can download. This is another version I've seen. This is, I think, what happens on Macs and on Linux. Then you download it into a file. And then once you're there, you go to apps. In App Manager, there's gonna be down somewhere here, Y files here. And then you're gonna have to install from file and point to the file that you downloaded. And it should do the same thing. I think this is difference between Mac and Windows. Linux is doing the same thing as Mac. So I've seen this thing before. So that's how it goes. And those Y files, every now and then they are going to send you a message that the license got to be renewed. So then you've got to reinstall it, and the problem should go away. So that's how it goes. Another. And the only way in practice, the only way to see what they do is to start with some toy network and see what happens if you apply one of them versus the other, and see what you like, what not. In many cases, making a more complex network is as complicated as splitting it into different subsections and then applying one of the layouts to subsection and then modifying it a little bit and then arranging those different modules by hand. So it's just repeated application of the layouts and manual creation. So that's how it goes. Any more questions on how to draw things in Cytoscape? Doesn't make sense. So if you want to practice, there's another file. So this is this polycom complex from the paper I got here. There's another file that I dropped in the on the Google Drive. This is it's not PMC, it was PRC. Is this guy. Okay, so where's my editor? File, open here. This is the same idea, but there's not. On Google Drive there are two papers. One is the one that I had the printout of. The other one is describing the structure of corresponding to the file here. This is complex number 3 or cytochrome BC complex from respiratory training yeast. So this is something that shows up in every dumb biochemistry class somewhere because this is part of the standard respiratory chain with several subunits. So you can play with this file, try to import it into Cytoscape as a network, then get it annotated by doing exactly the same thing as whatever I did with the PRC complex, and try to render it in some reasonable way. It should be easier. Yeast proteins are pretty easy to get data from UniProt. There should be all the stickers, everything should be coming in a reasonable way. So that's how it. This is something for you to practice, and I think 1 or 2 people do have some sort of a grading or whatever. They are taking this thing for credit. So then as assignment, so then I'm required to give you some sort of an assignment. So I'll come up with another file with another tiny network, so you'd have something to render and annotate the same way as you've seen here, and then I'll have to run you through a quiz, whatever based on whatever is going to happen tomorrow. So this is for those of you who do have assignment, do have this thing as a part of some credited class. So that's the formal side. So this is something for you to play with. And then, one more thing. Okay, so now you're not always starting with a network. Obviously, here I created toy networks from taken based on some protein structure, on complex structures. You might end up starting with the results of, let's say, some microarray experiment, which gave you a group of genes that is overexpressed under certain conditions, or group of genes that are, or group of proteins that were purified in some way because they are localized in some whatever. You essentially you end up with a list of proteins. Okay, you can get those things into Cytoscape even without having a network there. Okay, for this purpose, I'm gonna reuse the file I've got here, or not even that. I'm gonna reuse the file that I've got here. Okay, so let's save this thing. Let's start from scratch. So let's close it. And file, and let's import network from file, and let's pretend that this is my experimental data. Okay, let's open. How to read? Oh, because this is my compressed thing, so I've got to get the other one. Open this one. No, yeah. Oh, it hates tab. If it hates tab, then tough luck. Oh, UniProt, why not? File, import the table. No, import network from file. That's what I want. Import network from file. I want to get this. Yeah, okay, fine. So now, technically, I've got to specify where my each edge starts and where it ends, okay? But I can select none for import, okay? And then I can just select one of the columns to be a source node, okay? So I'm getting only a list of identifiers in my file as the starting node of the edge. I ignore everything else. Okay, if I do okay, the Cytoscape is gonna ask you if you really want to create the network with no edges, and if I say yes, I do have just a list of just the nodes with the proteins that I imported. Right, so this way you can start from a list of proteins and make it into network with no connections, just isolated nodes. Okay, so and then you can annotate those things the same way as we did it before. You use those identifiers to import stuff to UniProt, and this would work. Now, where to get edges from? And this is gonna be next 2 days, how to get it and what are the problems that you're getting in. Well, there's the panel here that lets you actually enter identifiers and then add edges, add interactions. But to do this thing, we need to type all those things here, or we can try to just go to node table, select everything, control copy, control V. So I just selected the values in a column and then pasted them here. This is a panel that talks to public databases that store information about protein-protein interactions, and you can query them directly from Cytoscape. Okay, and then there are going to return information what they found. So I just enter the list of those things. I click on search the network. And now you've got the panel showing up here, listing different interaction databases. There's quite a few of them. Some of them are functional, some of them are not. And they report you how many records are there for each of the database. Because of my personal connections and preferences, I do like IMAX. Okay, let's say that it's gonna come out why tomorrow, but that's what this is my go-to source of interactions if I've got to get it this way. There's gonna be 2,336 records there, okay? And then this is starting with a dozen of proteins. So whatever you're getting here can get large and can take a while to download. So it takes time. But we can try. Okay, we import. Okay, it will take a moment or more. The IMAX, the server is located in UK, so if there are problems with connectivity, whatever, if UK is, it can take time, but it should work. For larger networks, for larger number of records, it can take several minutes or whatever. So it's a time to get a cup of coffee and see what happens. But here we got stuff in. Okay. It asks you if you want to merge things. We don't want to merge anything because we've got only one data source. If we check more than one data source, that there would be some way of making those things into one network, but we do nothing. So we close. And here supposedly interactions that our have a dozen of, our dozen of proteins is participating in, or at least there's experiments supporting some sort of interactions here. As you can see, now we do have node table with a bunch of columns, way more than I got from UniProt. There's tons of stuff. Okay. Some of them are recognizable. IMAX is getting your identifiers corresponding to UniProt ID, so this is sort of convenient. It gives you taxon ID, so this is the same thing that we had from UniProt. We're getting a name of the species. We're getting a sticker which typically corresponds to a gene name, I think. You're gonna have way more stuff that you can handle. Some of this thing is just technicalities that are related to how the interaction records are kept track. So there's tons of stuff there, as you can see, but things that you don't want, you can always remove. Things that you don't want in a table, you can go to delete columns and then select things that you don't want, and you can toss it away. So this thing can be simplified. So this is on the side of the proteins, and then on the side of the edges, so interactions, it's even worse. Okay, you're getting tons and tons of stuff. Some of this thing is useful. Some of this thing is just the bookkeeping. Let's say you're gonna get information even when the original interaction record was created and when was updated for last time. So there's a lot of stuff that looks scary, but it's just everything that was in the record goes in there. So the easiest way to deal with this thing is to just pretend it's not there. Okay, so this is a relatively simple way of getting things in. Next step would be cleaning up the data, because if you check here, we're now gonna get strange things. Okay, we're gonna get, if we sort on, let's say, name, we're gonna get some of the identifiers corresponding to proteins, but then you're gonna have things that correspond to, as far as I can tell, genes. So those would be interactions between proteins and pieces of DNA. This might be chip or whatever data. IMAX might be reporting interactions other than between proteins. So this is one part to cleaning things up. There might be things that correspond to small molecules. Some databases within IMAX might report interactions between protein and small molecules. So those things might pop up. You will see things like here, like Unicode identifier dash something. This is related to splicing, and we'll go over this thing tomorrow, but there might be things like here that might require cleaning. If you check a column called now in the edge table, there's gonna be a column called interaction type or primary interaction type, and you're gonna have different types of interactions. This is related to the types of experiments and what information you're getting about interactions from experiments, and this is worth looking through. So this is just to signal what's gonna be tomorrow. Tomorrow we'll try to get to the stage that you'd be able to see things in imports from IMAX or from interaction databases and make sensible choices, what to keep, what to toss away, what is useful, what is not, how to make this thing cleaner than just a direct import. So that's gonna be for tomorrow. In meantime, you can play with this cytochrome BC1 complex to see, just to get. I mean, a lot of Cytoscape is just teaching your fingers to click on the right things to do those things in a more or less automatic way without much thinking. So the only way to do it is by clicking. So that's the reason. Okay, and I think that's it for today. If there's any questions, ask. If there's no questions, then I'm gonna try to stop the recording. Where's the recording? Okay, is there anything in chat? Nothing in chat. So I think I can kill this thing. I can stop the recording. We won't stop it.